Okay, so we now have a talk from the University of Liverpool, Dr. Leandro Xavier Neves, excuse me, pronunciation. Insights on regulated proteolysis of host and viral proteins in a time course infection of SARS-CoV-2.
Hello, I am Leandro. Please pretend everyone is not seeing murine or virus in there. I just forgot to delete. So I'm a postdoc in Ed Emmett's lab in Liverpool. Since I joined the lab, I've been using proteomics as a tool to characterize proteolysis in positive sensory RNA virus, of course, SARS-CoV-2. So proteolysis is very important for virus replication. We know that SARS-CoV encodes 2 proteases that are essential for polyprotein processing. But we believe that proteolysis goes beyond that. So we know that viral proteases can interact and also cleave cellular substrates, and there are some cellular proteases that can interact and cleave viral proteins. So we believe there is an interplay, a very complex interplay between different proteases and substrates, and we don't have a full picture of this yet. So we investigated the proteome of A549 cells infected with SARS-CoV-2 in a time course infection experiment and used mass spectrometry to detect peptides and infer cleavage sites on the proteins identified to understand enzymes and events, proteolytic events behind the cleavage we saw. The nucleoprotein, we detected a few cleavage sites. 2 of those are very interesting because they indicate a truncated form that is present in early time points of the infection, very likely to be a truncated form present in the virus stock we used. But at least for the second cleavage site, we see that splits the RNA bind domain from the C-terminus of the nucleocapsid protein. It starts getting very intense again in later time points of the infection, suggesting this is a cleavage site that's actually regulated in the time course. In addition to the nucleoprotein, we detected other cleavage sites in OF3, 7A, and OF8, but these are all mediated by the removal of signal peptides from these proteins. And we also detected lower levels of cleavage at the polyprotein. And these are known to be mediated by the virus 3CL protease. Can you hear me? Yes. In addition to the viral cleavage events we also detected cleavage at cellular proteins. We can see they are tightly regulated across the infection time course. They form multiple clusters, but most of these are mediated by cellular proteases, as we didn't see many of them matching the consensus motif that we know that N-Pro and 3CL and the PL-Pro proteases would cleave. But nevertheless, we believe these data sets rich in information of how the cell is responding to the presence of the virus during the infection. I'd like to thank our collaborators and the group, and if you're having any questions, please come to see my poster. It's number 11.